Hello students, welcome to the EPG Pathshala. I am Dr. Rajeshwari Sinha, working as Program Officer at the Center for Science and Environment, New Delhi. Today, we are going to learn about denaturation of enzymes. In this lecture, we will focus on understanding what is enzyme denaturation and how does it occur. We will then see what are the factors that influence the process of enzyme denaturation. This will include physical and chemical factors such as heat, radiation, mechanical agitation, acids and bases, ionic strength, organic solvents, cheotropic agents, etc. Then we will also look at the mechanism of enzyme denaturation. At the end, we will understand what is reversible and irreversible denaturation of enzymes. In today's lecture, we will focus on understanding what is enzyme denaturation and how it occurs. We will see what are the mechanisms involved in denaturation of an enzyme. At the end, we will see what do we understand by reversible and irreversible denaturation. This slide shows the concept map that will be followed in this lecture. First, we will see what is denaturation, how denaturation occurs, the various physical and chemical factors that influence denaturation, mechanisms of denaturation, and reversible versus irreversible denaturation. What is enzyme denaturation? An enzyme is a biological catalyst responsible for carrying out various biological processes within the living system. Enzymes work consistently until they are denatured, implying that when enzymes denature, they are no longer active and cannot function anymore. Denaturation of an enzyme is therefore defined as the loss of enzyme's biological activity due to some structural changes that are brought about in the protein by certain physical or chemical factors. Denaturation affects certain levels of the protein structure, which in turn also affects the orientation of the active site of the enzyme and interferes with the binding of the substrate to the active site. The proper enzyme function is therefore restricted. In this figure, we see two forms of an enzyme: the folded active enzyme, which upon denaturation becomes unfolded, and an inactive enzyme. In this section, we will understand how denaturation takes place at different levels of the protein structure. We all know that protein function is determined by all structural levels within the protein. During denaturation of enzyme, it is the secondary, tertiary and quaternary structures of the enzyme which are altered. This is because the bonding interactions which are prevalent in both secondary and tertiary structures are destroyed. The peptide bonds of the primary structure in the enzyme are left intact, since the denaturation reactions are not strong enough to break such bonds. Since the protein function is determined by structure levels of the protein can no longer perform its function once it has been denatured. In the secondary structure, denaturation disrupts the hydrogen bonding in the normal alpha helix and beta sheets in an enzyme and leads to unfolding of the enzyme structure. In the tertiary structure, the different types of bonding interactions such as hydrogen bonding, salt bridges, disulfide bonds undergo disruption during the process of denaturation. In the quaternary structure of the enzyme, the dissociation of the protein subunits takes place and the spatial arrangement of the protein subunits become disrupted during the denaturation process. Enzymes lose their biological activity also because changes in the enzyme structure levels further influence the orientation of the active site of the enzyme. Once the positioning of the active site is disturbed, the substrates will no longer be able to bind onto the enzyme active site. Further, the amino acid residues which are involved in stabilization of the substrate's transition states will also be no longer suitably positioned to do so. We have now reached midway into this lecture. In the previous sections, we have tried to understand what is enzyme denaturation and how it occurs. We also had a detailed look at the various factors influencing enzyme denaturation. We saw how pH, temperature, ionic strength, alcohol, metal ions and reducing agents act upon enzymes and denature them. We also saw some typical examples of these denaturing agents. Now, in the next section, we will try to understand what are reversible and irreversible enzymes. In this slide, we will see what are the various factors that influence the denaturation process in an enzyme. We will see that there are two types of factors, physical factor and chemical factors. The physical factors include factors such as heat, radiation, ultrasound, high pressure and mechanical agitation. Chemical factors include factors such as presence of acids and bases, high ionic strength, organic solvents, cheotropic agents, heavy metal salts or even detergents. There can be additional factors such as cross-linking, disulfide bond reducers, mechanical agitation, etc., which can contribute to the enzyme denaturation. We will look at the mechanism of enzyme denaturation by some of the major factors in the following section. Effect of pH. A change in the pH alters the stability of a protein structure and causes its denaturation. The pH of the enzyme solution can be altered by acids and bases which change the amount of H+ ion concentration in the solution. Protonation of the amino acid residues in the enzyme due to the binding of acidic proton to a nitrogen electron lone pair changes the way they participate in the process of hydrogen bonding, thereby influencing their biological function and activity. Not only this, acids and bases can also disrupt salt bridges between oppositely charged amino acid residues on the protein surface. Salt bridges are basically stabilizing ionic interactions in a protein or an enzyme. This is shown in the figure below. Salt bridges that exist between glutamic acid and lysine is disrupted in the presence of acid or base. Effect of temperature. High temperature often leads to weakening of the inherent bonds in the protein. Heat increases the kinetic energy and causes rapid vibration of the molecules to allow the bonds to get disrupted. Upon further heating, the hydrogen bonds within protein will be disrupted. New hydrogen bonds will be created within surrounding water molecules, thereby breaking the helical structure of the enzyme. Effect of high ionic strength. In dilute salt solutions, enzymes remain more soluble. This is because in their ionic form, the salts associate with opposite charges within the protein moiety. This leads to an increase in the hydration on the surface of the enzyme molecule. At very high salt concentration, the surface tension of the water molecules increase. This leads to a competition between the protein and salt ions for hydration. Salts therefore strip off the essential layer of water molecules from the surface of the enzyme, which is required for enzyme activity. Therefore, protein-protein hydrophobic interactions predominate, leading to the enzyme precipitating out of the solution. This process is also known as salting out. Effect of alcohols. The denaturation of proteins by alcohols or organic solvents proceeds through the disruption of the side chain intramolecular hydrogen bonding network in enzymes. This is simultaneously followed by the formation of newer hydrogen bonds between the new alcohol molecule and the side chain of the proteins. This is shown in the figure below. Water miscible organic solvents like methanol also tend to strip off the enzyme of its essential water layer. Hydrophobic interactions between enzyme or protein molecules thereby predominate, and the enzyme tends to precipitate out of solution as denatured enzymes. Presence of metal ions. In enzymes, heavy metal salts such as salts of mercury, cadmium, lead and silver are able to disrupt salt bridges. Upon reaction with a heavy metal salt with a protein, it usually leads to the formation of an insoluble metal protein salt. Heavy metals may also disrupt disulfide linkages within proteins due to their high affinity and attraction for sulfur, leading to an insoluble metal protein complex. Here we will look at the effect of reducing agents. Reducing agents can act on any sulfide bonds to break it. When reducing agents like mercaptoethanol are used for enzyme denaturation, they reduce the disulfide bonds present in the enzyme and convert it into the corresponding alcohol, which is the SH group. The figure below shows how a reducing agent acts on the cysteine moiety, denatures the enzyme and forms two different cysteine residues with SH groups. Effect of cheotropic agents. The commonly used cheotropic agents are urea and guanidinium hydrochloride, also summarized as GDMCl. Both have similar structure but differ in their ionic nature. Guanidinium hydrochloride is an ionic cheotropic agent which ionizes into GDM plus and chloride ions, thereby exerting cheotropic effects as well as influencing the stability of enzymes. Urea, on the other hand, is non-ionic and exerts only cheotropic effects. Examples of protein denaturants. So far, we saw the various types of factors which influence protein denaturation. Here, we will look at a few examples of proteins or enzyme denaturants. Acidic denaturants include acetic acid or trichloroacetic acid. Basic denaturants include sodium bicarbonate. Organic solvents may include ethanols or other alcohols. Cheotropic agents like urea and guanidinium hydrochloride. Cross-linking agents like formaldehyde and glutaraldehyde. Disulfide bond reducers such as mercaptoethanol and dithiothriatol. In this slide, we will look at the process of reversibility and irreversibility of enzyme denaturation. Reversible denaturation. When we say that denaturation is reversible, we mean that even after denaturation, the protein or enzyme can regain their native state upon removal of the denaturing influence. The process is also called renaturation. For example, an enzyme bovine ribonuclease contains 4 disulfide bonds. When treated with beta mercaptoethanol and urea, the disulfide bonds are reduced to SH groups, thus denaturing the enzyme and leading to the loss of enzyme activity. However, when the denaturing influence, that is the urea and beta mercaptoethanol is removed, the denatured ribonuclease slowly regains the enzymatic activity. This happens as the SH groups get gradually oxidized by the oxygen present in the air to form SS disulfide bonds. But there also remains a possibility that in this process of reoxidation of the SH groups, wrong disulfide bonds may form and the enzyme may not regain back its original activity. The resulting enzyme will therefore be termed as a scrambled ribonuclease. What do we mean by irreversible denaturation? When we say that denaturation is irreversible, we mean that after denaturation, the protein or enzyme will not be able to go back to its native state or renature itself even upon the removal of denaturing influence. For example, when egg albumin is heated, it denatures, resulting in the formation of a coagulated and opaque solid mass. This denaturation is irreversible and we cannot get back the starting transparent albumin protein even when the heat is removed. Students, we have now come to the end of this lecture. We have understood the concept of enzyme denaturation. Enzymes tend to work consistently until they are denatured, implying that when enzymes denature, they are no longer active and cannot function anymore. We have looked at what causes such denaturation and what are the mechanisms behind it. Let us do a brief summary of what we have learnt in this lecture. Enzyme denaturation refers to the loss in the enzyme's biological activity due to some structural changes that are brought about in the protein by certain physical or chemical factors. During enzyme denaturation, the secondary, tertiary and quaternary structures of the enzyme are altered. Primary structure of the enzymes are left intact. Factors influencing denaturation include physical and chemical factors such as heat, radiation, mechanical agitation, acids and bases, ionic strength, organic solvents and cheotropic agents. In a reversible denaturation, the protein or enzyme can regain their native state upon removal of the denaturing influence. On the other hand, in an irreversible denaturation process, the protein or enzyme is not able to go back to its native state or renature itself even upon the removal of the denaturing influence. Thank you for watching this lecture on enzyme denaturation.